Good afternoon, symposium members. It's always strange to give a presentation offline and then hear it back with you guys, but apparently it's one of the things that people do during a lockdown, so here we are. My name is Ferdinando Rodriguez and I'm a professor of medical robotics at Imperial College, and today, over the next 20 minutes or so, I'm gonna give you a bit of an introduction about needle steering for precision neurosurgery. This is a topic that has attracted a lot of my attention over the past decade or so, and I'd like to take you through the ups and downs of this journey and tell you a bit about where we're gonna go next. So first of all, I come from Imperial College, and at Imperial, robotics is a fairly big topic area. Over the past 4 years or so, we put together a grassroots initiative called the Robotics Forum, which basically brings all 40 odd academics in robotics under one roof, just so that we can work together, improve collaboration, and of course achieve critical mass. Now, what was interesting about this exercise is that over the course of putting together the Robotics Forum, we realized that Imperial is strong in robotics, and particularly it is strong around three main key areas: the home, robotics for the home, robotics for health, and robotics for infrastructure. Now, clearly I'm a medical roboticist, and so my topic area fits squarely within the healthcare domain, and so it is quite a nice moment to bring this bit of news to everybody's attention. That is that after a year of doing this role in an interim fashion, as of the 1st of July of this year, I'm now the engineering co-director of the Hammond Center. Now, the Hammond Center was set up almost 10 years ago to bring together healthcare robotics at the college, and of course it has a number of prongs that I will slowly familiarize myself with over the coming months and years. The center acts as an interdisciplinary research hub to facilitate collaborative research in the area of healthcare robotics, and it can do so because it has a wealth of state-of-the-art equipment and unique infrastructure that will basically give anybody working within the center a significant competitive advantage. But this is not the only thing that the center does. It has a significant and growing teaching curriculum with an MRes which spans across the faculties of medicine and engineering, and of course a number of summer and winter schools, plus the Hammond Symposium, which is probably the flagship conference for the Hammond Center. And finally, one of the key ambitions of the Hammond Center is to foster clinical translation, to help engineers within the faculty of engineering to find their more promising technologies and bring them all the way from the bench to the bedside. And of course it can do that because of the very strong and direct links that the center has within medicine. So that's a bit of an introduction about myself, but today I wanted to spend the next 15-20 minutes talking about a particular research area that has taken up quite a bit of my time over the past few years, and that is basically needle steering. Now, everybody in this space by now knows that surgery, surgical intervention has gone or is going from big open incisions to minimally invasive procedures. And of course that creates great opportunities in terms of, or great benefits in terms of shorter hospitalization, smaller scars, less risk to the patients, and so on. But with it, it also brings a number of important challenges, because of course as you work within a small or through a small incision, everything becomes more complicated. And so I guess a decade ago we began looking at ways in which we could do minimally invasive surgery within the head. And because the majority of instruments today remain straight, and so they are constrained to a straight line between an entry point on the skull to a deep-seated tissue target, our original fairly blue skies aim was to see whether we could develop technology that could allow us to steer along curvilinear trajectories within soft tissue of the brain. And the inspiration for this came from this particular insect. Is a wood-boring wasp, which a colleague and now friend from Bath University, whose name is Julian Vincent. He's one of the most prominent biomimeticists or zoologists in the country. What is special about this insect is the way that it lays eggs within the bark of a tree. And that is because the ovipositor, which is this long thin thing you can see here, is quite unique, as it allows the insects to burrow deep within the wood of the tree, even though the ovipositor itself is thin and long. And everybody with a slight engineering background will know that if you push something thin and long against something quite hard, you'll end up buckling or breaking that particular tube. The reason why it doesn't happen with the insect is because the tube is not actually one single structure, but it's actually a number of structures that are kept together by, you can imagine it as an interlock mechanisms. So you can see that in this little electron microscope picture here. Now if we synthesize why this mechanism is special, we can look at the little animation on the right hand side to see that the segments are moving reciprocally with respect to one another, and they've got this strange texturing on the outer surface. What this allows the insect to do is to burrow or push itself inside the bark of the tree, where the tensioning on one of the valves or the segments of the ovipositor is offset by the pushing force on the other valves. Now, of course, that particular mechanism has been then varied over a number of insects depending on the substrate that is being traversed. But the principle remains quite the same, that the tube is not one, but actually composed of many. Now, needless to say, when we first look at the design and try to copy it straight, we didn't get very far, and there is a whole other conversation about all of the pitfalls and all of the sort of dead ends we had to encounter in order to get a system that works. But basically, over a number of attempts, we eventually crystallized the essence of this design and ported it into a catheter type system. And what you see here is how this embodiment actually works. There are a couple of things here that are important for you to see. Now, there are no barbs on the outer surface, so the way that the needle moves within the tissue is thanks to the fact that at any point in time, 3/4 of this 4-segment needle are stationary and only one is moving. And because of this, you're basically holding onto the tissue with the stationary segments and pushing the 4th segment forward ahead of the others. And the second one, which we'll talk about in a little while, is that you can basically offset these segments one with respect to the other, and in doing so, you can actually define the orientation the needle tip takes as it traverses into the tissue. Now, strong of the basic results that we obtained with this early concept back in the early to mid 2000s, we sort of synthesized a basic understanding of why the needle works. And basically, it is diagrammatically illustrated in this presentation slide here. So if you imagine in a 2D world, there are no 4 segments, but there are 2. And the needle is pushed from the right to the left. So the needle, which is this one here, is pushed from the right to the left with an offset, which we call the steering offset, between the two. Now, if the brain is soft and the needle is flexible, as you push, if you push the needle from right to left, what will tend to happen is that the forces acting upon these two main bevels, the one on the top, the one protruding to the left and the one in the back, is not going to be the same. And because of this, you'll tend to see a bending of the forward protruding segment inwards, which in this case would be downwards. Now, what we could demonstrate is that the offset between these segments is actually proportional to the amount of steering the needle takes. And so these very early experiments can show you how that offset going from minus 30 millimeters one way to 30 millimeters the other way causes the needle to progressively change its radius of curvature from all the way bent in one direction to predominantly straight to bent in the other direction. And that was really step one. What we could also then demonstrate is that this was not only true in a plane, but it was true in full 3-dimensional space. Now, to do this, we had to do quite a bit of modeling and quite a bit of soul searching because it was nothing readily available in the literature. But finally, we came up with a control strategy which takes inspiration from the way submarines work. And so you can see here that by dynamically changing all 4 segments together, you can now pick any core orientation and any plane in order to achieve full freedom in three-dimensional space. And what you can see here is a quarter of our needle, where you can basically pick any curvature magnitude and any curvature plane and then steer right along it, where red is what we were aiming to do and blue is what we achieved. Now the second important advantage of this particular needle design has to do with the fact that because the needle segments are moving one with respect to the other, and at every point in time part of the needle is stationary and thus holding the tissue together, what we could demonstrate is that as the needle progresses, it deforms and strains the tissue a whole lot less than a needle of equal geometry that is pushed from one side to the other, as in conventional surgery. So what you see in this slide here, at the top, sees the needle pushed from left to right the classical way, and on the bottom, it is pushed reciprocally according to our biologically inspired mechanism. And without going through complicated statistics, it's quite obvious that if you look at the color code on the top, there is a lot more axial displacement, which is the displacement we care about, in the direction of the insertion compared to the bottom set of videos, which demonstrates that in the insertion process our needle tends to deform and thus strain the tissue less. And we published quite profusely on this topic to demonstrate that in fact the needle can be quite a bit less intrusive. Now, thanks to these very two advantages, the fact we could steer in full three-dimensional space and we could do so without having to twist the needle about its long axis, which is a great advantage if the tissue that you're traversing is as delicate and sensitive as the brain. And the second one is that because of this insertion process, which is reciprocal, we could drastically reduce the amount of tissue deformation and strain at the interface between the needle and the tissue. Now, strong of these two advantages, we were able to secure a fairly large grant, which is now 4 years in the making, which is called Eden 2020, and it's a Horizon 2020 partnership with a number of key stakeholders in Europe to produce a technology platform for neurosurgery. In a nutshell, we partner with a couple of companies which are sort of rather well versed in this area. So there's Renashaw, a large British corporation with one of the few neurosurgical robotic systems on the market, which has the name of Neuromate. Exograph, which is a commercial catheter extrusion manufacturer, which has been our chosen subcontractor to create medical grade steerable needles, and then clinical unit at University of Milano and San Raffaele to help us with the imaging and with the biomedical, so basically the neurosurgical application, Politecnico di Milano for biomedical engineering support, UMCG for shape sensing, and finally Munich or TUM for intraoperative ultrasound. And the idea was to basically spend 4 years together working on a technology platform that would allow us to do two things: therapy, and in this specific case is drug delivery in the brain, and in situ diagnostics, by basically using the fibers embedded within the needle to explore the possibility of doing Raman spectroscopy with the aid of the Raman team at Renashaw. Now, the system, back in the day where we were writing the proposal, was meant to look like this. It had a fine positioner, right, that held a bespoke trocar right at the entry point in the skull for the patient. That trocar then obviously holds a flexible catheter, our flexible catheter, which is biologically inspired. And then there is a second burr hole, which you can see here, which hosts a small transducer for intraoperative ultrasound. So that allows us to have eyes in the operating theater as the catheter is flexible and steers in the brain during a particular procedure. Then there is an in vivo diagnostic workstation, and of course a servo-assisted console, which is where the surgeon sits. This is a haptic joystick and a couple of monitors to basically monitor progress during the insertion process. Now, the system also had to come with some fairly advanced software. And the software had to do a couple of things. Allow the surgeon to plan on both MRI and CT what is the best insertion path for the needle to take to get to a deep-seated target. And then, of course, a way to identify the most optimal curvilinear path to reach that particular target that would get you there safely and, for example, avoiding major obstacles, so for example vessels or eloquent areas in the brain. And then, of course, intraoperatively we had to develop a completely new piece of software that allowed us to basically which would allow the surgeon to steer a bit like a three-dimensional cube, right, to steer the needle along a prescribed trajectory. Now, because we didn't want to start from scratch and we wanted the end output, the final output, to be of commercial quality, again we partnered with Renishaw, who already has a commercial planning and interventional software platform which is called the NeuroInspire, and basically endowed this software with additional functionality for both needle steering and the acquisition and processing of more complicated data, for example tractography and functional imaging within the brain. And so 4 years down the line, after some fairly tedious and difficult work, I am quite proud to show what the system looks like now. And I'm proud simply because usually when you have an artist impression of a system, it tends to then the implementation of it tends to be significantly different to what you had originally envisaged. But in this case, it actually is quite similar. So here you've got your fine positioner, which is the Renishaw Neuromate. You've got the actuators of our flexible catheter. We've got a flexible transmission that basically transfers these 4 linear motions from the actuators to the catheter. And here you see you get a sterile end effector with a single-use cartridge, out of which you get this 4-segment medical grade needle. If you then look on the left-hand side, you've got operational console with keyboard and mouse. There is a haptic joystick on the left-hand side, and of course a screen for navigation. If we take a closer look at the needle, this is the closest we have gotten to a commercial system. You now have a needle which is only 2.5 millimeter outer diameter, and it is carried within a sterile package, right? So you've got the needle within a cartridge and all of the plumbing that goes with it that sits within a single-use blister pack, which is sterilized, opened during surgery, and then disposed of at the end of the procedure. If we look at a cross-section of the catheter, you'll see that now in these 2.5 millimeters, you actually have 8 working channels. They are each 0.3 millimeters in diameter, and they're used predominantly for the insertion of fiber optics, for diagnostics and for shape sensing, and for the delivery of drugs to a particular tissue target. And then, of course, the software you see here, endowed with additional functionality, allows this catheter to follow, for example in this case, a curvilinear path that takes you from an entry point here to a deep-seated tissue target here, while avoiding the ventricles and the vessels. So where are we going next? Well, at this very moment, we've got a complete technology platform that has fairly unique planning ecosystem, a unique delivery and safe family of catheter for delivery of optics, diagnostics, and therapy to deep-seated targets within the brain, and of course a robotic navigation system to do this quickly and safely. And so with all these things together, we're now launching or beginning the process of spinning out a company called Neurosurgical that aims to take this technology platform and apply it to a number of important open challenges in minimally invasive surgery, among which we've got localized drug delivery for cancer therapy, in situ diagnostics, and of course things like therapy or tumor therapy using some sort of ablative mechanism. In this context, we already have a number of thriving relationships. One is with colleagues in Taiwan, with which we're doing in situ diagnostic using optical coherence tomography. We have promising work with a company which is outside of Oxford called Oxford Optronics, They do laser Doppler fluorometry, and we're using their technology to embed within our needles so that we can see ahead during the insertion process, identify that there is a vessel in the way, as to avoid it and avoid hemorrhages. We have the beginning of a relationship with a French company called Servicio, who do optical biopsy for again in situ diagnostics. And finally, we have a continued relationship with Rana Shaw and their Raman team. So with this, I would want to conclude by saying, of course, this is not just my doing, but it's a fairly extensive team effort with a lot of people that have contributed with their PhD and their postdocs over a number of years. And of course, it is also rather expensive technology, which started with funding from EPSRC, continued with funding from an ERC starting grant, and then of course is now fully funded by a European Horizon 2020 grant, which is a Research and Innovation Act. And so I guess this is the end of my pre-recorded talk, but I'm also here, I suppose, listening to these same 20 minutes, and I look forward to answering any questions. If you need to find me, of course, that's my email address on the bottom left-hand corner, and you can, of course, find me on various other social media. Thank you very much.